Okay, I think I'm recording. Yeah, I'm recording. Hello everyone, or whoever's watching this. Welcome to my project where I will be characterizing the microbial community associated with facial skin and different areas of facial skin. All right, so let's get started with some goals for the project. So our main goal of this project was to describe the microbial community that is culturable and it has been previously associated with different things. I know everything was different for everyone, but for me, it was the microbial community that is associated with facial skin. And I think another important goal of this project was to sort of experience and understand the steps that must be taken to start with a sample or an idea of what you would like to sample and grow in a lab setting, enrich the conditions for it, and ultimately sequence it and come to an assumed conclusion for yourself in your own project. So I think that was a really important goal here. Okay, so let's get started with some background of the skin as a structure as of itself. So the basic purpose of the skin is to serve as a physical barrier to protect our internal environment from foreign polluted substances, as well as work to maintain our body temperature and prevent fluid loss. We have sweat glands in our skin that release urea when we sweat in order to cool down our body and as well as maintain the heat in our body in colder temperatures. So the skin's a large organ, we all know that. It's about 1.8 meters squared in size, and within that 1.82, or within that 1.8 meters, there's a magnitude of different environments. We have, sweat glands, as I previously said, hair follicles, folds, vaginations, creases, and each of those different sort of aspects of the skin can facilitate the colonization and the growing of a vast variety of microorganisms. So that's why there is so much diversity in the microbiome of the skin. The three layers of the skin, epidermis, dermis, and hypodermis, not too important to what we're talking about here, but just thought it would be sort of reasonable to insert this picture right here, where we can see right up here, the skin surface is where we're seeing the most of the viruses, bacteria, and fungi, and the skin surface, as well as seeping down into the epidermis. But we can also see that they can reside in the sweat glands, sebaceous glands, which are just like oily glands, and the hair shafts as well. So the community of bacteria or virus in the sweat glands is obviously going to be very different than what is on the surface. That's going to be a much drier environment. All right, so now let's get into it as an ecosystem, as sort of like a home to all of these microorganisms that I'm talking about. So generally, the skin is pretty cool and acidic, about 4.7 in pH, and dry. Most of the areas of our skin are very dry, not in the type of dry that we think like dry, flaky skin, just dry in climate. There's not a lot of moisture, there's not a lot of oil, except in specialized areas, which we'll get to. And we see a symbiotic relationship between the organism's skin and the microorganisms that colonize on those skins. We give them things that they need to survive, like, and an example I have here, some organisms will use the urea that we produce in our sweat as a source of food. And they work to protect us and to sort of facilitate the environment that is on our skin. So as I was saying, the communities are very variable depending on the environment they're in. So in a sebaceous, oily environment, that's where we're going to see the lowest diversity of microbes, like the lipophilic residents, such as propionion bacteria. And then we have our moisture areas. Oh, backtrack a little bit. One example of like an oily area that I could give you would be like the crease of your nose is a pretty oily area. I believe like the back of your neck would be a very oily area just because of the fallout from your natural hair oils that it naturally produced. But then we go into moist areas, which is home to the aerobic organisms, who I said previously probably use the urea and sweat as a nitrogen source. These are organisms such as Staphylococcus and Crinum bacteria. That's where they reside. And then we have our dry areas, which is actually the most diverse site for the microbiome of our skin. And in a couple different literature, they cited that it was actually more diverse than the gut microbiome. So we know that the gut microbiome is very diverse. There's a lot of different bacteria there that are working in a symbiotic relationship with our gut to metabolize these foods that we're eating and absorb nutrients properly. It's sort of the same situation on your skin as well. But the dry areas is where we'll see home to most of the gram-negative bacteria, along with phylum such as actinobacteria, proteobacteria, firmicutes, which is bacillote as well, that phylum, and bacteroidetes. So as you can imagine, there's a lot of different external variables that can affect your skin's composition. Your age, you have more wrinkles, more folds. That's a different environment than a flat surface forehead. If there's wrinkles there, it's going to be a different composition than if there wasn't. Along with obviously climatic factors, temperature and pollutants in the air, and just all of that sort of stuff can definitely interact with the microbiome of your skin, causing, to inhibit some microorganisms or facilitate the growing of some others. So there's definitely a lot of variation that comes into play when looking at what is actually growing on our skin, but there has sort of been like a baseline of what has been seen a lot in literature. Alrighty, so my project, so I wanted to do the facial skin because I know that there's different kinds of environments, different, some oily parts and some dry parts, some moist parts. And from like research that I did previously, I knew that Staphylococcus was a very, very common skin dweller that has been cited in multiple different literatures. That's been sort of like the hype around like skin microbiome is a lot of coccus bacteria or gram-positive bacteria as well. So I decided to do a culture and to grow on mannitol salt agar due to the high salt concentration that selects for gram-positive bacteria like Staphylococci. I know that Staphylococci can handle very high saline compositions. I believe the saline of this is about, sorry, I have it in here somewhere. Oh, it's about 7.5 to 10% NaCl. So that's a pretty high salt concentration. That would be inhibitory to most gram-negative bacteria. But this also differentiates between strains of bacteria that can ferment mannitol that results in a pH change and ultimately a color change. So when the bacteria can ferment the mannitol, they'll produce an acidic byproduct, which will change the pH of the agar, resulting in it to change color as well. And it also differentiates between pathogenic and non-pathogenic Staphylococci rather, sorry. Pathogenic will degrade mannitol and use it as an energy source, and non-pathogenic strains will not do this. So to begin my project, I swabbed 6 different areas of my skin because of the difference in environments of them, and grew them all in mannitol salt, all in the same conditions, 26 degrees Celsius in the incubator. So this one right here is the cheek. This one is the corner of my mouth that is comes in contact with a lot of foreign substances, food, drink, you're touching it, a straw, multiple different things come in contact with that mouth area. The under eye right here comes in contact with a lot of tears, which are antimicrobial, which we've learned through this class. So I thought that would be kind of interesting. We have the nose crease, very oily, hairline, very moist from sweat. And then the eyelid, which I was pretty interested in the eyelid to begin with, because when you have your eyes closed, there's not much of a fold, but when they're open, there's a fold in your eyelid, and it can tend to be pretty oily there as well. So, you have sort of an alternating environment of a fold being there and then not being there when your eyes are closed, like when you're sleeping. So I wanted to see if, that had anything to do with what we could be seeing in this culture. So I continued to culture. I moved forward with the eyelid sample just because of the interest and because it did not have a pH change. It stayed the color red rather than turning yellow like the other ones did. So from that original sample that was in the previous slide, I collected a colony from that eyelid plate and then again streak plated onto a new mannitol salt agar. Also let that grow in the same conditions in the incubator. And this first subculture also, I did not see a pH change. So I'm guessing that there was sort of a very mixed community and there was not enough bacteria to ferment this mannitol and produce that acidic byproduct to see a pH change. But as you can see, as I continue to subculture, this was my second subculture, definitely a stage up from that very pink color, working towards that yellow color, which we see here in subculture 3. This is the most sort of bright yellow, distinct colony, looking like it should be sequence culture that I had. This one was a step back. I'm not too sure what happened with this one, but this was from my third subculture. And this one right here was seemingly the most pure to me. So I collected a colony of unknown community, placed in a test tube, and that was sent to UNH to be sequenced for its DNA. All right, now let's get into the meat, the taxonomic classification. So the sequence data that we were provided with by Patrick software. And so let's start sort of broad schema, we'll work our way in. For domain-wise, we had 92% bacteria hits, 0.1% eukaryote hits, 0.02% virus hits, 0.01 archaea hits, and 8% no hits. Now, I was a little surprised by these results. I knew I was going to get a good amount of bacteria, but I also thought that I was going to get a much higher percentage of eukaryote DNA because I was taking samples straight from my skin and just streak plating it right on. So I assumed that, you know, I would be seeing some of my DNA in there as well, but 0.1%, that's a pretty low percentile of eukaryotic DNA. All right, so let's narrow in a little bit on that 92% of bacteria that we saw. So from that, we've got some Firmicutes/Bacillote phyla, that's 91% of the bacteria was that. 0.5% of the phyla was proteobacteria, and 0.05% was of the phyla Bacteriodota. The rest of the sort of percentage makes up a bunch of bacteria with very low percentile, and they were all gram-negative. I'm not too sure if that was sort of like a contamination reason, or if maybe, you know, I just had some like byproducts of walking to class, soil, pollutants, whatever on my face, but they were mostly gram-negative, the smaller percentages. All right, so now let's narrow in on the phyla. The major phyla we had, the Firmicutes/Bacillota. Of that 91%, 56% of it was Lysini bacillus, and 34% of it was Staphylococci. Both gram-positive bacterias, which is sort of what I expected. I definitely expected a higher percentile of Staphylococci in comparison to Lysine bacilli, but you know. So the major players of Lysine bacilli. So we had the species Ferricus, fusiformis, and species YS11, and their percentiles are listed here. I was really surprised by the extremely large percentile of Lysine bacillus I had. I did not see it cited in much literature when doing my research for this project. And in my most pure culture, the one that I, gram stained and did the phase contrast microscopy on, I was not seeing rod-shaped cells, which is what would be seen with a Lysine bacillus culture. So I definitely think that maybe this was in higher proportions when we had sort of the pinky color that we saw in the beginning, and like the less pure subcultures, because these do not degrade carbohydrate sugars. So these will not metabolize that mannitol, and they will not cause a pH change. So when we're seeing a lot of these in a sample, we will not see that pH change on the mannitol salt agar. So I'm guessing that there was a lot more of these, and we sort of cultured them away as we moved on. Major players of Staphylococci, species-wise, we had some Mornier, Capatis, Pasteuri, Epidermis, Auroris. Percentages are listed here as well. These three strains up at the top are pathogenic strains. That's what I'm assuming I had in my most pure culture, because once again, of the fermentation of the mannitol, only the pathogenic ones do, and non-pathogenic do not. So I'm seeing very low percentile of the non-pathogenic strains. So that sort of is in agreement with my results, if you may. All right, so for the characterization, this is the gram stain that was produced using the most pure culture that I made. Samples were collected with a sterile loop and placed on a glass slide. Typical gram staining procedure was followed. As you can see by the picture, I have gram-positive bacterial cells by the purple color. And if you just look a little bit closely, they look like a bunch of little grapes, circular grapes. So that is the coca shape that we would see in a Staphylococci sample. Phase contrast microscopy sort of confirmed this for me. So yeah, my conclusion would be that even though I did go into this expecting Staphylococci, I enriched the conditions to grow Staphylococci. And I assume that is the community that was seen in my purest culture, maybe a multitude of species in there. But as far as genus goes, I assumed, and I assume I do see Staphylococci. But I was rather interested in the discovery of the large amounts of Lysine bacillus on my skin. I only sort of saw this cited as a skin dweller in a handful of literature. One in particular was an article that was written, I believe, in 2019. That was done by, I believe it was done in China, and it was done on 2 different age groups, 20 to 30 and 50 to 60. And in the 20 to 30, they saw a very high amount of proteobacteria. On the 50 to 60, they saw a lot of predominantly the Licin bacillus, which I thought was quite interesting and sort of got me thinking, okay, well, maybe is this sort of where we are seeing the external variation, the age coming into play on changing the microbiome of our skin. So I thought that was pretty relevant. And I think that this whole sort of broad scheme thing is very relevant when it comes to skincare. Skincare is a very emerging field, especially the biochemistry of it, the working with the microbiome to produce better results, to, you know, whether it be anti-aging or hyperpigmentation. There's a lot of sort of contingency between the microbiome and the formulation of these products, the ingredients of them. And medical procedures as well. If you're, you know, getting surgery and you're not, there's a lot of, you think that there's a lot of Staphylococcus in your body and you're being prepped to deal with that, to inhibit that bacteria. But what is happening is not, per se, inhibiting Licin bacillus, and that gets into your bloodstream, that can cause infection. So I think it's really important that we know that Licin bacillus can be a massive skin dweller. So I think that there definitely has to be some more experimentation done on this. And as I just said, further experimenting can be done to enrich the conditions to grow Licin bacillus rather than Staphylococci, so that you can sort of, you know, gram stain and phase contrast to select for Licin bacillus rather than Staphylococci. So that's my presentation. And here are my references and acknowledgements. Thank you.